Hello, and welcome to the first in our series of major metabolic pathways in cell biology. In this series, we are going to give an introduction to energy in general, but then we are going to focus more specifically on cellular respiration. In our next series on metabolism, we will discuss alternative pathways to cellular respiration, as well as another major metabolic pathway, photosynthesis. But in this series of lectures, we're going to discuss energy transformation in cells, the role enzymes play in this process. We will discuss cellular energy in the form of ATP, the major mechanisms of its generation, and then we'll move on to discuss cellular respiration, which is one of the major energy transforming pathways in cells. All right, let's go! First, it is important to understand that we have two states of energy, either usable energy or stored energy. I like to think of it kind of like money. Either you're spending it or you're saving it. Energy that is being stored or saved is potential energy. Sort of like money in the bank, this is an energy reserve. It can be used whenever needed. Kinetic energy, on the other hand, is readily accessible energy. Think of this like money or energy you're using in a transaction like cash or card. Typically, kinetic energy is referred to as the energy of motion, while potential energy is stored energy. In order to understand this at the cellular level, we need to zoom back in really closely in our organization of life, like back down to the atomic level. Recall that electrons, which are the basis of chemical bonds, or in other words, the building blocks of everything, are not part of the atomic nucleus. They kind of swirl around the atomic nucleus constantly. They are not static. This means that electrons are always zooming around and moving. Even when two atoms form a covalent bond, they're still kind of swirling around, although they're spending their time kind of around two atomic nuclei instead of just one. This means that electrons are pretty much the perfect example of kinetic energy. They are the energy of motion. When they have the space to zoom around an atom, they have the most kinetic energy. Atoms in general also have a certain amount of energy associated with them, in part because of the kinetic energy associated with electrons. However, when atoms form bonds, the sharing of electrons actually reduces the amount of kinetic energy associated with the atom because it sort of restricts the space that the electrons have to zoom around. For this reason, when electrons are shared in the form of bonds, they are said to be stored there. So this would make chemical bonds a rich source of potential energy. If these bonds were broken, the energy would be released as kinetic energy. I mention this because as we move forward, you need to start thinking this way. If electrons are released from a bond via that bond breaking, that is usable or kinetic energy. If electrons are being used to form a bond or being stored in a bond, they're essentially stored in that bond as potential energy. Another important source of potential energy in cells are concentration gradients. Recall from our discussion of membrane transport that concentrations occur when molecules are in different concentrations on either side of a membrane. Recall that molecules want to be in equilibrium across the membrane, or basically in equivalent concentrations. So having a different or higher concentration on one side of a membrane establishes a sort of pressure in the sort of a gradient. This is potential energy. Think about it this way. Here is a membrane. Here you see a concentration gradient of hydrogen ions. There are more hydrogen ions on this side of the membrane compared to this side of the membrane. These ions are being stored on this part of the membrane. So think about it like a dam, like the Hoover Dam, shown here. That dam is storing a tremendous amount of water on this side of the dam. That water holds a tremendous amount of potential energy. I mean, think about what would happen if that dam broke. All that water would come rushing through. aka the energy of motion, a lot of kinetic energy. Concentration gradients are the same. When a lot of molecules are stored on one side of the membrane, it's essentially like damming a river. Tons of energy is stored. We can use that energy to fuel reactions. As you can see here, and as we will discuss later, concentration gradients store so much energy, they're actually one of the most efficient ways to generate enough energy to create a high energy bond between adenosine diphosphate, or ADP, and another phosphate to create adenosine triphosphate, or ATP, the cell's preferred source of energy. Before we move on, it's important to understand a little bit more about energy. The study of energy, its states, and the transformation of these states is called thermodynamics. Essentially, how do we go from kinetic to potential energy and potential to kinetic energy? And what happens when we do? For this course, there are two relevant laws of thermodynamics. First, energy is constant. It cannot be created or destroyed. This is important to understand because it means that we have a constant amount of energy in the universe. We cannot create more energy. We have what we have, and we have had it from the beginning of the universe. How wild is that? Atoms make up matter, and all of the matter in the universe has existed from the beginning of time. I mean, take a minute to think about that. That means that all the electrons and the atoms that make up your body were once part of dinosaurs, were once part of the very first organisms on the planet, were once part of the very stuff that was created at the beginning of time. I mean, wow. Okay. Wow. That is a lot to take in. Okay, anyway, back to the present. The fact that energy is constant just means that we can't make energy for use. We can only transform it. So we can transform potential or stored energy into kinetic or usable energy, or vice versa. This highlights the importance of metabolism. So metabolic reactions are what enable the transformation of energy. They enable us to release energy stored in chemical bonds for use of other chemical reactions, or they enable us to create bonds in order to store energy for use at a later time. The second law of thermodynamics says that this transformation of energy is not necessarily 100% efficient. Energy can be stored relatively efficiently or transferred from kinetic to potential, but when energy is transferred from potential to kinetic, this involves the loss of heat. Heat energy is actually kinetic energy, but it's just not energy that can be used in a chemical reaction. So it's said to be lost. You can observe this pretty easily if you think about driving your car, which involves using gasoline, which is a large molecule composed of a lot of carbon-hydrogen bonds. When you drive this car, these bonds are broken to release electrons, which can be used to energize the car. However, once you stop driving, if you were to put your hand on the hood of the car, it would be hot. This is because when those bonds were broken, that energy was released, heat was released as well. This is similar to when you use glucose to fuel the production of energy in the form of ATP, or when you're using energy to fuel another reaction, like muscle contractions when you're working out. Think about hopping on the treadmill and going for a jog. You're breaking the bonds in ATP to fuel muscle contractions. But when you're done, how do you feel? You feel hot, like heat hot, but also hopefully confident. These two laws are important to remember because when we discuss metabolism, or how cellular reactions take place, we need to understand that we are discussing the transformation of energy, not its creation. As I mentioned, metabolism is the sum of all energy transformations in a cell, the transformation from kinetic energy to potential energy and vice versa. Thus, we can broadly characterize, first, recall that kinetic energy is basically free electrons, while potential energy is the storage of these electrons in chemical bonds. Therefore, we can broadly characterize all metabolic reactions into two types: reactions that store electrons in chemical bonds and therefore build bonds between molecules and are using small molecules to generate bigger ones. These type of reactions are called anabolic reactions. These require energy input because, well, you have to have money to put it in the bank in order to store it. Energy has to be put into these reactions because these reactions are involved in storage of energy in chemical bonds. Again, that is typically referring to storing electrons in chemical bonds, but we can also use energy in the form of active transport to generate a concentration gradient, which is another robust form of cellular potential energy. When energy is required as an input for a reaction, the reaction is said to be endergonic. On the flip side, we have catabolic reactions. These reactions involve breaking bonds in larger molecules, resulting in smaller molecules. When bonds are broken, energy is released. This essentially means that electrons were involved in or stored in a chemical bond and are now free to form bonds with other molecules, so they've been released. When bonds are broken and energy is released, this is a transformation of potential to kinetic energy. Recall that these reactions also generate heat. When energy is released during a reaction, the reaction is said to be exergonic. Okay, so anabolic reactions are those that require energy input to build large molecules from smaller molecules. Catabolic reactions are those that require energy via the breakdown of larger molecules into smaller molecules. All right. This is a good time to remember back on when we discussed macromolecules, especially since macromolecules are the substrates of the majority of cells' metabolic reactions. Recall that two primary types of metabolic reactions involved in the creation of degradation of macromolecules were dehydration synthesis and hydrolysis. Dehydration synthesis is when a bond is formed between two monomers to form a polymer. For example, when glucose monomers are linked to form starch, cellulose, glycogen, etc. Or for example, when amino acids are linked together to form polypeptides or proteins. Since bonds are being created between monomers leading to the production of polymers, dehydration synthesis reactions are anabolic reactions. Since the sharing of electrons is involved in the formation of these bonds, electrons are stored in these bonds, making these transformations of kinetic to potential energy, and therefore endergonic reactions. Hydrolysis reactions break bonds in polymeric molecules, release individual monomeric subunits. For example, when polysaccharides such as cellulose, glycogen, or starch are broken down into individual glucose subunits, or how polysaccharides or proteins, polypeptides or proteins are broken apart by proteasomes into amino acids. Since bonds in polymeric molecules are broken, leading to the release of individual monomers, hydrolysis reactions are catabolic reactions. Because this would release or free electrons from being stored in these bonds, thus enabling their energy to be used elsewhere, these are exergonic reactions. Again, heat is also released during these reactions. But wait, we're discussing some pretty powerful exchanges of energy here, like building and breaking bonds. How do we make sure that these exchanges of energy occur when we need them to? Well, remember what macromolecular group catalyzes chemical reactions? Yeah, buddy! Enzymes catalyze chemical reactions. You're correct. Most of these chemical reactions need a little encouragement to occur, and if you recall, that's the function of enzymes. The amount of energy required to enable a reaction to take place is called the activation energy. This is the amount of energy required to either build or break a bond. And usually the amount of energy required for a reaction to take place is quite high, meaning that it is unlikely that the reaction will take place spontaneously. Thus, enzymes help speed up chemical reactions by lowering the amount of energy required to enable a reaction to take place. For the vast majority of reactions we discuss moving forward, there are corresponding enzymes that enable them to take place in a timely and regulated fashion. Another thing to note about metabolism in general is that metabolic reactions like cellular respiration or photosynthesis or polymerization and more are multi-step processes that involve several linked reactions and enzymes. These are called metabolic pathways, and in many cases, these are self-regulated in a pretty cool way by what is called feedback inhibition. This diagram does a really good job of showing a metabolic pathway and feedback inhibition. Imagine that the overall goal of a certain metabolic pathway is to convert molecule A into molecule D. This involves a series of 3 changes, 1, 2, 3. And these changes might be like 3 bonds being broken or 3 bonds being created, whatever it is. What happens is reaction 1, which is facilitated by enzyme 1, converts A to B. Then B is converted to C by reaction 2, which is catalyzed by enzyme 2. C is then converted to D by a reaction facilitated by enzyme 3. Success! We have made tons of our product. But as we convert A to D, this means we're running low on molecule A, which may be a very important molecule. So we want to slow this process down so that we don't run out of A by converting it all to D. So what we can do, and what happens in many metabolic pathways, is the process of feedback inhibition. And this is when the final product, in this case product D, is actually an inhibitor of the enzyme that converts A to B. So therefore, A is converted to D through a series of steps, but once we get enough of this product, it actually is used to turn off this pathway to prevent any further conversions of A to D. So from this lecture, you need to focus on understanding the major sources of cellular energy. So focus on electrons and chemical bonds and concentration gradients especially. You need to understand the first and second laws of thermodynamics and how they apply to metabolism. You need to be able to compare and contrast anabolic and catabolic, as well as exergonic and endergonic reactions. And you need to understand how feedback inhibition can regulate metabolic pathways. You need to be able to use the terms potential energy, kinetic energy, thermodynamics, anabolism, catabolism, endergonic, exergonic, and feedback inhibition. Thank you so much for watching this lecture, and we look forward to you hearing the rest of our metabolism lectures.